Now let's move on to the program. So now this is my great pleasure to invite Dr. Swapan Datta, the DDG Crop Science in Indian Council of Agriculture Research. And this country, India, has been immensely benefited by his able guidance. So please join me in
Good morning to everyone. I think you all agree that Rajiv personally set up this program in such a manner that everyone will feel that they know nothing if they see the galaxy of the scientists sitting there. And that's a big credit, I should say, that Rajiv personally has organized this meeting with all the best possible scientists in genomics that are here today. Of course, there's a great leadership from Dr. William Dahl, the PhD of ICAR. He knows how to get the best possible things done. That's his leadership, and that's the way things are moving at ICAR, the best possible way. Dr. P.K. Gupta, he's our guru in genomics in Bangalore, and it's difficult that to say things that when he's there, but he's very constructive, criticizing person. I always appreciate his critics. And of course, Dr. Van Zoon, that will be giving the inaugural lectures. We all know that Beijing Genomic Institution's contributions, and one joins that what he will be talking today. We're all excited to listen to him. I see lots of people that I have been associated for a long time. That here, Dr. Gary Aplin, many other ED colleagues. I will not go on naming all the names. My also colleagues of ICAR, also Dr. Siddiq, Dr. Arthur Sharma. My 1985-86 association when I was working with Benson, Andreas Graner, he's here. So I see lots of people that I know, some of them, Robert Pettigrew is a good friend of me. And they're all here, so it will be next 3 days with an exciting meeting. And I'm sure all will enjoy this meeting. I was coming from a meeting in Ahmedabad. There are 600 plus Indian seed industry people are attending that meeting. There are 50 foreign seed companies representatives also at present. And there are discussions on how to move the seed industry in India as well as beyond India, and how to use the technology and getting good quality seeds and its availability. Now I see this particular meeting, which is basically genomics breeding, genomic-based seed development. This is absolutely befitting that this is the science and technology that would drive the best quality seed development. What I actually mentioned in my speech there, that science and technology, one difficult thing that it takes time. It takes time today. What science will be doing, it might take 10, 15 years time to see the product. But the science that being generated or being conceptualized long back, the product we see today, and we must utilize those products. What I said, from integrated farming system to integrated genomics breeding. And that's the whole concept that should emerge from this 3 days meeting in the long run. We now know that since long back, when this particular genomic science emerged, there's a digital revolution took place in this particular development. Now we all know that the value of genotyping, the value that genotyping to phenotyping, dissecting the traits and loci and the markers associated with those kind of loci and the traits, and how to pyramid those markers, how to pyramid the allele mining, ultimately to develop the designer crops that would be utilized in the farming system. Now, meanwhile, definitely, I'm sure Dr. Andreas Graner will be talking on high density genotyping, which is very important. From the very base to when you go to the more in-depth, and gradually these all things will come. Similarly with the DART sequencing, it was 15 years back this was developed. I'm sure now we would know that where we have moved since it has been reported, how this can be utilized further in our breeding program. There's another issue that with the climate change and other reasons too, the low yielding environments where all the stress, biotic and abiotic stress are involved, how we can utilize this genomics linked plant breeding, developing the new material, looking for the new loci, new markers that should be linked to those traits and utilize them. It's not that easy because we recently, last 2 years, 22,000 of wheat germplasm and 18,000 of chickpea germplasm have been evaluated at different environments to look into or to screen the new genotype or to screen certain genotype or trait. We're finding great difficulties. Of course, we see some exciting message, but we also see great difficulties in linking the physiological manifestation and genetic characterization of those material. It might take what we thought very simple. We will find out straightforward some material and look into that material for its dissection. But it was not that easy, and it might take even longer for phenotyping. Rather, to me, it seems that genotyping probably is moving faster than the phenotyping, and the challenges still remain with the phenotyping, and that one big challenges need to be done. On this contest, I would appreciate and thank Rajiv, because one of the projects that Ministry of Agriculture and USAID led project, what I missed on 18, the review was there, and I'm familiar with the progress that Rajiv Adikli said made, that they're linked to the phenotype. All those the draft sequencing data available to us, and those are being utilized along with bioinformatics, they're being utilized for phenotyping. Entire country of India as well as other partners are involved in this particular program, and I think this is one massive program is moving in, at least in pigeon and chickpea, and I wish many of the crop plants, they take the same pathway, the linking genotypes with the phenotyping. And definitely it is possible today. The next generation genome sequencing allows us to look for the whole genome sequencing, and definitely that would facilitate the identity of the loci. And once we know that, the remaining part would be simple. Now, a few things that what I learned from the seed meeting, that certain other traits that normally we discuss in plant breeding today, that we need shortening the cycle of the breeding, one thing. Second, we need shorter duration of the maturity of the plants, because today is not high density genotyping, the high density cropping patterns, many of the other things, architecture of the plant, they're all required for mechanization, required to reduce the disease incidence, etc., etc. And for that, this patterns is very important to identify and to know it. As quick as we understand those markers, as quick as we can identify those genes, encoding those characters, and we can put together, I think that would make a remarkable progress in course of the future plant breeding. Now the last questions that I have, based on the omics data that we have today, or continuously emerging, can we predict the phenotype? Because I see that our present generation of the scientists, the young scientists, they don't want to go to the field. They don't want to be very much in the field. Earlier we thought this is the right thing to do it. Except Shifa Liang's lab in China. Shifa, as a good thing, one thing he made it, maybe some other labs also in China. I know more Shifa, that's always referred over Shifa. That in the morning, 7 o'clock until lunch, all the scientists, the majority of the scientists have to work in the field. They're not allowed to come to the lab or to do any work in the lab. After lunch, they come to the lab where they do the molecular biology. And in the night, probably they link how the molecular biology can be linked to the phenotyping. I wish in India we can do these things happen, but it's not happening. Now we must look the science-based understanding of the phenotyping. Can we predict in the laboratory? Can we predict all the omics data, either it is genomes or transcriptomes or methylomes, and along with the bioinformatics tools available to us, can we correctly or at least towards correction the phenotyping, and then can link in a limited greenhouse glasshouse condition primary phenotypings, then extend this material and information to the actual field. That probably would accelerate the breeding program, would generate new excitement in young scientists. They can be connected to the field work. Ultimately, we would be able to integrate the genomics in what we are looking for. Thank you very much.
Thank you very much, sir, for your guidance on the integrated crop improvement programs.